Okay, yeah.
Okay, we're live. Brilliant. Okay, that means we have to start. Excellent. Hi, everybody. I'm really glad to be working with the Chemical and Systems Biology division for doing this presentation today. I realize that we're just at the very start of the Systems Biology unit. You've had your intro yesterday, I think, from Jonathan Blackburn. He is a really great scientist. I've really enjoyed working with him. I'm usually over here visiting his lab every Tuesday. My schedule's been so chaotic lately, it's been hard to do that. But he asked me if I'd be willing to teach as part of this honors program, and I'm really glad for the chance. So I'm glad to meet you all. I am presenting the first of 8 lectures that we do for the Bioinformatics module at my own division. So this is a slight update on what I've done a couple times before. So today we're going to talk about data properties and kind of that interface where computer science ends up touching systems biology, because there are an awful lot of computer science concepts that show up, and we just sort of assume people know about them, but in fact they don't. So today we're going to be covering several of these areas. We're going to start with this topic of metadata. Metadata is one of these topics that everyone insists we need more of, but you frequently find that people don't make a very good description of what metadata is all about. So we'll try to cover that at the start. From there, we're going to talk about numerical representations. Again, we assume that a lot of people know what binary is all about and what it's useful for, but today we're going to talk about how binary representations lie behind the way that we report image data and the way that we report character data as well. From there, we're going to move to a discussion of compression, a topic that shows up all over the place in bioinformatics, in large part because our data are really huge. So we need to have efficient ways to store that information so that we don't just flood every hard drive that gets touched by a bioinformatics project. And from there, we'll talk about more complex data stores than just simple text files or Excel. There are a lot of things you can do in Excel, no question, but certainly Excel is not the answer to every person who's got a data storage problem. So we're going to try to talk about reasons why we might want to move to, say, XML as a data storage method or relational databases. And then finally, we'll take a quick tour of high-throughput sequencing from a perspective of the file formats that fuel it. We're going to start with some discussion of the pipelines that we frequently see bioinformatics described as supporting. And from there, we'll move to talk about high-throughput sequencing and just looking at different file formats that we use for storing information of different types. All right, any questions before we move ahead? Well, if other people show up, we'll just tuck them into seats along the way. So, all right. Well, you're all sitting all the way at the back, so I'm going to stand about halfway through the room. That's fine. So we're going to start with metadata. Metadata is frequently poorly defined as data about data. That's what meta means. It's kind of at the next level up. So if you write a tweet about social media, you're doing meta social media. If you have data that instruct you what data mean, you have metadata. So we could start with just the basic explanation that they are experimental descriptions. They explain what the experiment was about. They contextualize the results, though. Contextualize means that we have these data that came from something, but we need to understand the context in which they were produced to understand them. So metadata make it more possible to understand what your experiment was all about. We would also note that one of the things we always brag about in science is that we make reproducible findings. That, for example, if you publish a paper, you look kind of silly, frankly, if somebody else tries to follow exactly the steps that you did and they don't get the same result. Metadata are pretty key to that. If you write a very basic paper about your experiment and leave out a lot of important details like what software you use to produce a de novo assembly or what statistical system you use to produce your p-values, it's going to be really hard to interpret that. I mean, if you just see a p-value in a paper, you kind of want to know what tests they use. You kind of want to know what assumptions lie behind the statistical model they use. So metadata, in a lot of ways, it enables this replication process. They are key to the reproducibility of science. I also want to point out that an experiment without metadata is wasted effort. And that's a pretty high charge to lay, but I want you to keep in mind that the amount of money made available to scientists and the amount of time available to scientists certainly are very limited. The government is not suddenly going to double the amount of money flowing into biomedical research. We're not suddenly going to have double the number of people to be able to make sense of our experiments. So if you're going to publish an experiment, you need to do a very careful job of documenting it through metadata so that people can understand that result well. I've included some links at the bottom of these. Some of these are direct URLs like this one. You can just click on that link and you should be able to get anywhere you want. But you may also see me give peer-reviewed literature down here. In each case, that's intended to give you access to deeper information about the topic. They're not, I don't consider everything I cite at the bottom to be something that would be covered by quiz questions I put together on these. So output files alone are not all the data you need to understand an experiment. Frequently, the experiments we do, supported by Bioinformatics, are working with core facilities, like maybe we work with CPGR across the way. We send them some samples and back comes the data. So you might think that the data files contain everything you need to understand them. But I want to point out that frequently to the people running these core facilities, what you've given them is sample vial this, that, and the other, and they don't know what your experimental structure is. So I want to imagine that you're a new graduate student and you have been bequeathed data. There was a postdoc in the lab a couple of years ago, and he or she produced this data set, and you've now inherited it as a graduate student. So I want you to imagine that what you see is a file called DT vial 26, DT vial 93, DT vial 05, vial 59, vial 23, vial 85. What you've got, if you've just got those 6 files, is nothing. You have no idea what the context of these. And so you might end up scouring through this postdoc's lab notebook, hopefully they kept a lab notebook, to figure out what the heck these 6 files represent. If they, on the other hand, noted that vial 26 maps to biological replicate 1, that's promising. Vial 93 to biological replicate 2. Now you know the kind of relationship that these 2 files should bear to each other. You know more than if one vial were sent to the facility and they were told to analyze it 3 times, where you have technical replicates rather than biological. Hopefully, you also know that these have been separated into 2 different cohorts. Maybe a TNF alpha treated cohort and maybe a control cohort that's had some sort of mock treatment. So having the ability to layer an experimental hierarchy onto your output files is one of the most basic kinds of metadata that we can ask. A lot of times you will see that people have nominally fulfilled their requirement to make their data openly available by posting it to a repository, but they didn't produce something that says which file belongs where in the hierarchy. So metadata are critical for that. I also wanted to note that metadata is something that you do every day. When you post a photo to Facebook, one of the things that it will frequently do is ask you, who's in this picture? It'll give you a face and say, is this person your friend Jill? Or is this person your friend Jack? So these are, this is an example of, the example of Facebook image tagging is an example of adding metadata to a bitmap, a JPEG image in this case. So the other thing is that every time you take a photo with a decent camera or even with your cell phone, you are collecting huge amounts of metadata about that. How many of you have location services turned on your phone right now? Almost everybody. I always leave it turned off because I'm a paranoid freak. But one of the things that your camera in your phone is tagging on each photo is a type of data called EXIF. EXIF is an example of metadata that we use for describing media files. So JPEGs are one example of that. So I was over at UWC the other day, and I took a picture of my campus, that's Tigerberg Hospital, Tigerberg Hill behind it. I thought it was a great image, what can I say? And I took a look at what metadata were tagged within that image. And what we see over here is a whole pile of information. It knows what its file name is. It knows how big it is. It knows that I collected this on my Canon EOS M2. I know exactly which version of firmware it was using, what the shooting date was, I don't have a GPS on this camera, or it would have tagged the exact coordinates I was standing at that date. And it even records what kind of lens I was shooting with, what the ISO settings were. All of that is in there. And if you shoot it on your phone with location services enabled, it's going to write down the date of that. So let us say that you take a photo of someone opening presents at Christmas time. If you have location services turned on, your phone has captured the coordinates at which you celebrated Christmas. Maybe you just report, and you post that to Facebook. Suddenly, Facebook has access to the exact location you were standing on Earth when you opened Christmas presents. Maybe they know where mom and dad live now. So metadata are important also from this perspective of privacy and security. A lot of people use systems like Foursquare to check in to say, I'm at the gym, and they post that to Facebook. Now Facebook knows where you go to exercise, and people can track your movements based on an explicit release of metadata by you. So have a little bit of paranoia, protect yourself, it's a very wise thing. I never post a photo with location data from my home, or anybody else's home. All right, this brings us to minimal information standards. So a lot of people in a lot of fields have grown really tired of people publishing incomplete assessments of their experiments. And so minimal information standards are one of the efforts that have come out of this. So this is a way to create a canonical set of information that should be expected of anyone publishing in a particular systems biology field. So in this case, we have the standards for the minimal information about a microarray experiment. This is pronounced Miami. I'm an American, so that comes naturally, right? But there are also standards for publishing in genomics, or publication standards in the world of proteomics, for that matter. So don't expect that just because you're not working with microarrays, there's no minimal information document that guides you in what kinds of information you should post. So I'm just going to read this quote taken from this down at the bottom. This is a post from the European Bioinformatics Institute. It's a very valuable site to know about for accessing all kinds of tools in bioinformatics, and training, for that matter, in bioinformatics as well. So minimum information standards are sets of guidelines and formats for reporting data derived by specific high-throughput methods, sequencing, microarrays, whatever. Their purpose is to ensure the data generated by these methods can be easily verified, analyzed, and interpreted by the wider scientific community. You, as soon-to-be grad students, are already probably thinking about the projects that you're going to be engaged in. You want those projects to have the greatest impact that they possibly can. And a study that is a textbook example of how to report data sometimes gets cited just as an example of how to do it. So being somebody who follows Miami and Miyagi and MIAPI standards gives you a much better chance for your data to be cited as a great example of how to do this kind of research. Sometimes this happens unintentionally. One of the first papers I wrote when I was at Vanderbilt, in collaboration with a group downstairs, used canine cells, dog cells, as a way to study basement membranes. And one of the things that we did was to use RefSeq as a database for identifying our proteins. And this was highlighted as a data set of the week by one of the repositories, simply because we had demonstrated a way that you can use a genome-only derived protein database for identification in non-model organisms. Dogs are not that frequently used as model systems. So I was really pleased. We got a lot of extra publicity on our paper just because of how we had structured the database search. Okay, with that, we're going to move to a little bit of math. I'm sure this is going to be very exciting. Now, I have to say, the other group I taught this lecture to had just had lunch, and they were sleepy. So I'm sure that you're going to follow this a whole lot better. We're going to talk about binary. Everyone knows that computers are digital, that they store information in ones and zeros, but very few people have any concept of what that actually means. So today we're going to talk about bitmaps and how they relate to the numbers underscoring them. So at the moment, let's just pay attention to this grid over at the left side. I've written in a series of powers of 2. 2 to the 0, 2 to the first, 2 to the second, 2 to the third, all the way up to 2 to the 7th. So how many columns do I have here?
7 and 8.
8, right. That's a good error to make because I want you to be thinking about this. Computer scientists frequently start counting from 0. So when you see 2 to the 0th through 2 to the 7th, remember it's 8 items, not 7. Great. So 2 to the 0th is the same as anything to the 0th power. Anything raised to the 0th power is? 1. Great. So if we have this final bit turned on, then we're going to add 1 to the total. 2 to the first is? 2. There it is. So anytime we have this bit turned on, we're going to add 2 to the total. 2 to the second is 4, 2 to the third is 8, and so on. If that bit is turned on, we're going to add that corresponding power to the number. This representation allows us to represent any decimal number by a sum of bits. Okay? So in this case, we're going to ignore sign. We're only going to think about 0 and positive numbers. Those natural numbers, I can't remember what you call the set of numbers starting from 0 and including only positive integers. It's all right. We'll stick with natural numbers mostly. So here we are, if we have 8 zeros in a byte being, in this case, 8 bits, we have 0, 0, 0, 0, 0, 0, 0, 0. That means that we've got no additions. The total is 0. If we have all of those bits turned on, we get the number 255. So our range for a byte in unsigned and an unsigned byte is from 0 to 255. There are 256 different values we can achieve. All right, so let's count them off. If we want to represent 1 decimal in binary, we just need to turn that final bit on. So 0, 0, 0, 0, 0, 0, 0, 1. That's binary in this case for the value of 1. The binary value, and if you had to write that with just the only significant bits in place, you would just say 1 binary is 1 decimal. To represent 2 decimal in binary, well, we can't make this last digit 2, can we? Because we only have 2 possible values at this location, 0 or 1, which means we have to go 1 significant figure higher. So 1, 0 is the same thing as 2 in decimal. If we turn on both of the last 2 bits, we add 2 to 1, and that gives us a 3 decimal. So one way to read this figure, then, is simply to count them off. If you count from 1 to 8 in binary, you get 1, 10, 11, 100, 101, 110, 111, 1,000. So when we reach 1,000 in binary, we've only reached 8 in decimal. Clear to everyone? Okay, so this is the first part, to understand how collections of bits allow us to represent larger numbers. When we deal with characters in most computer programming languages, we're not dealing with Unicode or anything complex like that. We're just trying to represent, say, English text. We're going to stick with 1 byte representing each character. We're going to talk about how we do that in a few slides. When we deal with a short unsigned integer, we have 2 bytes, which means we have how many bits? We said 16. We have 16, exactly. We have 8 bits per byte. So with 2 bytes, we have 16 bits, which means that instead of having a maximum value of 255, we have a maximum value of 65,535. So that's an important thing to think. By having 2 bytes glommed together, we don't have 256 plus 256 numbers, different levels that we can count through. We have 256 times 256. So 65,536 different levels that we can report with just 2 bytes. Frequently, we use long integers in our programming because sometimes you need to count a very large number of events, for example. And for that, you would use 4 bytes, which means that you would have 65,536 times 65,536 different values you can report. Now, I've noted up here, oh, we've got our projector flipping to yellow again. That bothers me so much. I had this fixed.
Yeah.
Give it another poke, give it another kick. No, I think the projector is just like that. I'm sorry. All right, so what I've been talking about so far is characters and integers only, and I haven't slipped in this problem of dealing with sign. What's your sign, right? So we need to have the ability to flip these to be able to handle negative values as well as positive values. In effect, we integrate the sign with the magnitude values here. So if you were to report a short integer, we said that there were 65,000 different levels you could have. If you are using a signed number instead, you can no longer get up to 65,535. You can only get up to 37,768 and down to a similar amount negatively. So we have to basically steal a bit in order to do that. So when we go to signed numbers, we have what's called 2's complement to do this. All right. Now, when we deal with float numbers, things get really complex. How do you represent pi with a bunch of binary digits? The answer is that we have the number that, when we do scientific notation, how about Avogadro's number? Anyone remember Avogadro's number, what it's for? Avogadro.
It's like to calculate moles, I think.
Yes, very good, yeah. So when you want to calculate how many atoms are in a mole of some element, Avogadro's number is the answer. Anyone remember the number?
6.2.
All right. So I remember 6.02.
Yeah.
6.02. Okay, 6.02 times 10 to the 23rd.
Yeah.
So this is an example of scientific notation. So we would write that 6.02 E23 if we're being really compact about it. So in giving Avogadro's number that way, we're splitting it into a notation that computers are quite happy with, because we have some number always given as only one significant figure past the decimal base, to the left of the decimal. So we have this number between 0 and 10 that is being multiplied by 10 raised to some power. Everyone get that? Okay, 6.02 is the number to the left. E just says that whatever follows this is the power to which we are raising 10. 10 to the 23rd in this case. So if we were to represent that in a computer, we would use a bit for, I believe this is the sign of the exponent, so 10 to the 23rd, so that's going to be positive in the case of Avogadro's number. We have, let me try to remember this, we have 8 here representing anything from negative 127 to positive 127. So that's going to be 23 in the case of Avogadro's number. And we have 23 bits left over just to describe that part that comes to the left of the E in the scientific notation. So when we do a floating point or a double precision floating point value, we have to split up the bits that we're using. So 1 plus 8 plus 23, what's that come to? How many total bits?
That's 32.
32. 32 bits is how many bytes?
4.
Exactly. So we use the same amount of storage. Oh, we're back to a proper color. That's good. I did flip again. My vision is kids playing bloody tricks with this. Okay, so the amount of storage we use for a long integer is the same as what we use for a standard float in C, for example. And sometimes you want to have all the precision you can possibly muster, and for that you would use a double float. So 1 plus 11 plus 52 bits, I believe that comes to 64 bits. So we can use 8 bytes to store a very high precision floating point number with a wider range than what we can achieve with just a single float. All right, so that's a start to understanding how numbers are represented in computers. I realize it's kind of complex, but I think it's valuable to think about the fact that the amount of information you're recording about a character or a number is going to be reflected in the amount of bits that has to be allocated to it. Okay, but wait, there's more. We're not just representing numbers, we want those numbers to mean something. So I want you to think about what we call the RGB notation for storing information in bitmaps. Okay, so now we are going to have 24 bits for every pixel of the screen. right? My computer has the ability for all of these little dots on screen to project red and green and blue. Any questions so far? Did I move too quickly or about right there? Okay, that's good. So I want you to recall that our computers have screens that can show us all kinds of different colors. Now the way that they do it is a little shifty. If you had a lobster looking at the screen, the lobster would see different colors than we do. It's not the case that every dot of your screen can appear any one color. In fact, every one dot has 3 different colors that are being shown in different ratios to you. Sneaky, right? So, our eyes have receptors that are good for measuring brightness and for measuring red and green and blue. So, if the computer is able to project those colors at us, we perceive the cumulative sum of those 3 colors. So in effect, each image that we project, that we have on a computer, is represented by 3 bitmaps behind the scenes. 1 for red, 1 for green, and 1 for blue. So how do you get yellow out of that? Well, what we see right here in Mr. Projector, if I whack it enough, does it go white again? Our projector is showing in yellow today, and that's because it has 3 projection colors, red, green, and blue. And the blue is on the fritz. So if I thump it enough, maybe the blue comes back on and this looks white again. But because blue is missing from this white background, you're seeing yellow. Squirrely how eye color works, isn't it? So we have 3 planes then that are represented in these 24 bits. The first 8 bits represent the amount of red that we have. The second 8 bits represent the amount of green that we have. And the last 8 bits represent the amount of blue that we have. Except Mr. Projector is ignoring the blue bits right now. So you can think of those as just having numbers from 0 to 255 for each of those colors at each position. So when you buy a camera, would you touch anything below, say, 10 megapixels? 10 megapixels good or bad? Okay, we got an AV snob over here. Anyone acceptable on 10 megapixel? When I first moved here, I bought a really cruddy telephone, like the worst thing you can get on the summer specials for running Android, right? And it had a 1.3 megapixel camera in it. People looked stressed when I said 1.3 megapixel, right? So that means that there are 1.3 million picture elements here, and each of them has 3 bytes being recorded for it. And with my camera, which is my Canon EOS M2, I have an 18 megapixel camera, so 18 million dots in this space. 3,000 some by 5,000 some dots. At each dot, I'm capturing 3 bytes, red, green, blue. Okay, so if you have 255, 255, 255, how would you decode that for the color of the dot? Well, what does that, if you've got 255, 255, 255, what is the, do we have a maximum or the minimum intensity for each of red, green, and blue? That's the maximum. 255 is the highest value. So you have the maximum amount of red, the maximum amount of green, the maximum amount of blue. That's white. It's a it's the brightest that any one dot can be, pure white. Now, what if I had 128, 128, 128?
It's half the intensity.
Half the intensity on all 3 channels. So that's going to be kind of a muddy gray. All right? So now let's go the other way. What if we have 255, 0, 0? What am I representing? What's that? One color. Which one was it? Red, yes. So the order is red, green, blue. So if you have 255, 0, 0, you have a really bright, very saturated red. If you have 255, 255, 0, you have a very bright yellow, kind of like what we're seeing on screen here, if this were any brighter. So that's combinations of these 3 colors give us every color that the human eye can see, more or less. I'm fudging a little bit on that. So this is how our computer screens and how our cameras fake color for us to see. Okay. How would that change if we reduced the number of bits given to each of those three colors? So let's start at the very worst. Let us imagine that we use only one bit to represent each dot in a picture. This is what we call, well, we would call it a line art black and white image, meaning that every dot in that image was either fully black or fully white. There's no gray allowed for there. So a 1-bit representation squeezes out all the shades of gray, squeezes out all the color. We just have that 1 bit. Next, you might have 2 bits for each channel. So this doubles the number of shades you can represent. You've got black, dark gray, light gray, and white. Those 4 are all represented when we have 2 bits to spare. When we have 3 bits, we can now cover 8 different shades of gray. 4 bits get us up to 16, 32, 64. By the time you get to 6 bits, the intensity information that you're seeing starts to look photorealistic. And we found that about 256 different levels of intensity help us, are essentially, we're at that stage of indiscernibility that you wouldn't notice a huge amount of difference between, say, 192 and 191. They're very, very similar to each other by our perceptions. So this is one of the reasons why when we do 24-bit representations of color, we usually limit ourselves to just 8 bits for any one channel. If you use a really high-end camera, it's probably storing internally more than 8 bits. Because it may be that you're shooting in a very dark room, or maybe you're shooting on Table Mountain with the sun glaring right on you. The camera needs to be able to deal with all of that intensity. So this tells us about how the mappings we use, the number of bits we use to represent something, give us different levels of accuracy in reproducing it. All right, and we should talk just a little bit about number bases. We've already been doing some of this when we talked about binary and decimal. Decimal is a base 10 number system, probably because we have 10 fingers. Binary is a 2-bit system, which is great when you're dealing with digital computers and you've got transistors that are passing or not passing current. So here we have 3 different number systems all thrown together. Before we counted from 1 to 8 in decimal, and I had that grid to show you 1, 10, 11, 100, 101, 110, 111, and 1000. So those are the binary representations. Frequently, because we work so commonly with 2-byte systems, or 1-byte systems for that, in this case, we think of hexadecimal, base 16, as another valuable number system to work with. So imagine if, a moment ago, when I was saying, what if I gave you, we were talking about red, green, blue color triplets. I asked you, what if I gave you 255, 0, 0? And you said that was red. We could also have said, what if I gave you FF, 0, 0, 0, 0? This is a way that we can represent 255 or 256 different levels in each with just 2 characters, not 3. Hexadecimal is a way of giving binary in sort of a shorthand form. Trying to trot around these really long strings of 1s and 0s is just not that tractable. So it's useful if instead we can compact that to a number system that uses 0 through F as numeral symbols, not just 0 through 9. So let's count together to 16 in hexadecimal. All right? We will, so we're just going to work our way through that. So we start with 0, 0, 0, 1, 0, 2, 0, 3, 0, 4, 0, 5, 0, 6, 0, 7, 0, 8, 0, 9, 0A, 0B, 0C, 0D, 0E, 0F, and 10. Right, 10 base 16 is the same as 16 base 10. Just one of those weird realities of life. Okay, so when you see someone representing numbers using letters, just keep in mind that A comes after 9. It's a little like when you're playing card games. You know, when I really enjoyed playing spades when I was a teenager. We played it every time we were out on a competition in high school. spades all the way there, spades all the way back. So when I was playing a card, I knew that if somebody had laid out the 9 of diamonds, I could beat that with the jack of diamonds. So in this way, the jack is used to represent the number after 10. It's weird. So that's somewhat, in the same way, we can use A to follow 9 in order to produce hexadecimal. Okay. So let's then talk about how we map to something that isn't really a numerical value, but still needs to be represented as a number for us to be able to deal with it in computers. And that gives us ASCII. So ASCII is really old at this point, and yet it is held up pretty darn well. If you have a Roman alphabet language, you can represent it using ASCII. So here we have a hexadecimal table. This is one of the reasons I brought up hexadecimal when I did. We have our numbers from 0 to F, and here we just have 0 to 7. So are we showing, how many different values are we showing in this table? How many rows are there?
17.
There are 16 in this case, because we don't go all the way up to 10. So 16 rows, and we have how many columns? 8. So 8 times 16 is?
It's, wait, it's coming. 104. 104.
No. Last digit.
16 times 8. It's either 104 or 102.
128, very good. All right, so 128 is a special number in the world of 1 byte because?
It's almost half.
Exactly, it's half of all the different values you can represent. 256 values may be represented, and in the first half of them, we can represent the characters. So, for reasons I will not get into, A, the capital A, starts at letter 65. So in this table, we know that we can multiply 4 by 16, because it's in the 4th column, and add 1, because it's in the first row, the 1 row, and that gives us A, where that shows up. So anytime 65 appears in a number sequence, we can instead read that as the letter A. Anytime 66 appears, we can read that as the capital letter B. If we wanted to show lowercase, we would need to slide over here. So here we need to add 32 to A to get to the lowercase a. All right, so this has been the standard way for representing letters with single bytes in computers for a very, very long time. There are also systems called EBCDIC and so on that are also built around 1 byte systems, but generally speaking, everyone speaks ASCII at this point. Now, you might be asking, what happened to the other 128 values, right? We had 1 byte, so why didn't we use the last 128 levels of that byte? The answer is we did. We have some additional standards like things called ANSI that allow us to represent all these accented characters. Anyone have an accented character in your name? Anybody in the room? Oh, okay. Well, I frequently see people with, say, an E with an acute accent over it. So you can represent those with some of the characters beyond 128. Okay. So that's the ASCII table. ASCII is really valuable to know, to at least know what it represents, because if you see a series of numbers in something and want to understand what they represent, especially if you're doing anything cryptography, you're going to be using ASCII every day. Okay. So this brings us to the fine topic of compression. Who has ever used compression on your computer? 3, 4?
Yeah.
Several people have. Actually, everybody has. I'm just going to be really clear. If you've ever worked with an image file, like a JPEG, you've worked with compression. If someone has ever sent you a zip file, an email, you've dealt with compression. Compression is everywhere, but how does it work? Well, today you get to find out a couple of the basic ways anyway. So, biological data are big. We very, very frequently deal with data from sequencers, for example. And when we deal with data from sequencers, we definitely want to compress it because we don't want to lose our entire hard drive to the single experiment we put on it. So we must use compression to fit data in as small a space as we possibly can. So is it just when I'm standing on this cable that...
Yeah.
Aha! Let me see if I get it fixed. Only that once. How about over there? No? No? All right, fine. We'll deal with it. Fight with a projector. So we want to, ideally, use the smallest number of bits possible to give us a faithful representation of the original content. Now I'm going to stretch that definition a little bit. In a couple of slides we're going to talk about lossy compression. In lossy compression, we do not demand the ability to recreate the original perfectly. So there are a couple places where that's very valuable. We're going to talk about that. So you may see that you have a file that doesn't say a .zip extension or a .rar extension. That does not mean it's not using compression internally. So many files will compress little bits of things internally. When one of the little secrets I learned along the way making these presentations was that formats like PPTX and DOCX and XLSX are actually compressed files. If you have a Word document on your computer, you can save in the DOCX format. You can create a copy of that and change the file extension to .zip. Immediately you can look at all of the different inclusions in there, like all the images that have been attached as figures inside that document. So compression is used all over the place, and you may not know it. So if you see these file formats, I would suggest that you keep in the back of your mind, that's a zip format. Frankly, the inclusion of a Z is a pretty good giveaway that you're dealing with a compressed file. So .zip is the PKZip format. .gz is the gzip format, typically from Linux. BZ2 is the block sorting 2 format from Linux. And .rar is, oh my goodness, I forgot what that's called. Remote archive, something like that. So you'll frequently see files that are called .tar.gz. The gz is the compression part that's being applied. Tar is just conglomerating a whole pile of files into one. So tar is not itself inherently compressed. I've included a URL to 7zip.org. If you ever receive a file on your Windows computer, at least, that is zipped and you don't know how to decompress it, 7zip is a really powerful utility. It's much faster than the one built into Windows. Okay, so we're going to start with one of the most basic forms of compression that we use. It's called run length encoding. So I want you to imagine that we have an image that is 1 bit deep. Right? So we said that in a 1-bit image, where each dot is represented by a single bit, we usually think of that as just a black and white image. Each dot is either black or white. So in something like that, you will frequently see long stretches of just white dots. So something like that should be really open to high levels of compression. And run length encoding is the answer for that. So here we have a string of letters. We have AAA, AAA, AA, BBB, BBB, CC. Obviously a bunch of repeats, right? And this is not an example from black and white dots, but we've got 3 symbols, and each of them is appearing in a straight run. So run length encoding is able to reduce this stream of bytes into A8, B6, C2. To represent that A showed up 8 times in a row, B showed up 6 times in a row, and C showed up twice. So how did we do with our compression? How many bytes did we have in our original stream?
16.
16. We had 16 characters in a row. And now, how many bytes are we using to store that? 6. That's pretty good. So if you can get that kind of compression out of run length encoding, you're in a very happy place. That's very helpful when you have a very small number of symbols that are showing up in runs. So you could imagine that if you had sequencing data that might compress very well by run length encoding, or it might compress very badly. So imagine that you have a short tandem repeat, something that has, say, CA, 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 CA. What's the run length in that? Just 1. No character is repeated right after itself. So for something like that, if we're able to recognize that the symbol that's repeating is 2 characters, then we're going to be fine to be able to compress it. So the level of abstraction at which runs are being detected would matter. So that's clearly not the only way that we want to be able to do compression. Certainly if you have something like a grayscale image where you have all 256 shades represented, suddenly your ability to find runs is very limited because we have a large number of symbols. 256 different symbols. So let's think about English for a moment. I borrowed this URL for the Concise Oxford Dictionary to ask simply, how many times does each letter appear throughout that dictionary? Just counting the words themselves. Now, I note that this is not the same thing as asking how frequent is each letter in the English language. And the reason for that is that we use some words a lot more frequently than others. I mean, what was the last time any of us used the word milk toast? Milk toast. Milk toast. Don't let anyone ever describe you as a milk toast. It's spelled with a Q, by the way. Now you all keep going milk toast. That's fine. Okay, so here we have our list of all of the letters as they appear in the Concise Oxford Dictionary. And we see that the most common letter is E. Well, we all knew that. Now see, in the United States, we all watched in the 80s anyway, we all watched the game show Wheel of Fortune. And you could offer your letter when it was your turn, and they would flip those letters over in the phrase you were trying to guess if the letter were represented. If the letter weren't represented, you lost your turn. So it didn't make a lot of sense for you to say, show me the Q's, unless you were really bloody sure a Q showed up there somewhere. Okay, so we see that Q is all the way over here at the tail. It's very much the least used letter. But it's in pretty good company. It's got J and Z and X. All four of those letters, though, are pretty darn rare. So we have to go 2 letters in before we get to letter 3, the R, that is the 3rd most common letter in the English language. Okay, so Huffman Codes work on the principle that we want to use fewer bits for symbols that are more frequent. Okay? So if we have to use more bits to represent a Q, that's not so bad, so long as we can represent E with fewer bits. All right, so it is a greedy algorithm. In this case, it is the algorithm for producing a Huffman code. I'll flip back for just a moment. Is to look at the 2 symbols that have the least occurrence and join them together. So we start with this list of 26 letters. Then we have 25 letters, with the 25th letter being a combination of J and Q. And you keep replacing the 2 least frequently seen items into a new item at each stage. So what are the 4 letters that are least common here? X, Z, J, Q. When we look at our Huffman code that derives from this, we see that X and Z have been merged to create a letter called XZ. We have J and Q that have been merged to create a symbol called JQ. And we see that those 4 letters are joined to create JQXZ. All right? E, on the other hand, is way up here. E was the last letter merged in because it was it was the most frequent of the individual letters. That said, it's not merged in at the end. Instead, the ADF set is merged in with the BCE set at the last stage. So how can we read this? How does this turn into a compression format? Imagine if each letter is represented by potentially a different number of bits, and we no longer have to specify where we are at the end of this letter. So the magical bit is that if you have if you see the string 0, 1, 0, that corresponds to a left, right, left in this graph. So left, right, left, 0, 1, 0 corresponds to the letter E. If we want to so that is 3 bits that we use to represent E. How many bits does it take to get down to one of these rarest of letters? We need to go left, 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 right, 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 for example, to get to Z in that case. So that's a total of 1, 2, 3, 4, 5, 6, 7, 8 bits just to get to Z. And yet, on average, if you are trying to encode a particular letter or a particular word from the Concise Oxford English Dictionary, this will dramatically reduce the number of bits that you use to represent that whole word. So let's do the word the, T-H-E. Easy, right? So where's T? Anyone see it?
I see it.
Right here. Okay, so to write the word the, we're going to say 1, 0, 1, 1. And then to that we add 0, 1, 1, 1, 0. And finally, 0, 1, 0. Do we have to use any spaces? We don't. Because every time we hit a leaf node, every time we hit one of the final terminating letters here, we know that the next bit has to start at the top. So we can store the word the very, very compactly just by knowing what the frequencies of letters were in the dictionary from which it was derived. There are some words that will not be optimal from this. For example, the word quiz, Q-U-I-Z. Q already takes 8 bits, we know that. U takes 1, 2, 3, 4, 5 bits. I is a pretty good one actually. That takes 1, 2, 3, 4 bits. And Z again takes 8 bits. So quiz is a word that falls some distance from the standard uses of letters in the dictionary. Therefore, it's going to take more bits to represent, a disproportionate number of bits to represent, because its frequencies are so strange. Okay, so that's Huffman encoding. It is a standard method that we use for compressing data. Now, I mentioned that I was going to break the mold a little bit. We are, in this case, not going to make a perfect representation of the object we're compressing, and we're going to use something called lossy compression. Lossy is very, very valuable, and it's one of the reasons why you can do video on your phone. It's one of the reasons you can do audio on your phone. Lossy compression means we can throw away a little bit of the least important information, the parts that people are least likely to notice, and have a much more compacted file as a result. Has anyone here, I'm going to ask you about copyright theft for just a moment. Has anyone here ever ripped a CD, made MP3s from a CD?
I have allegedly.
Someone has alleged this about you. I hear that. All right. MP3s are not as big as CDs. A CD is approximately 650 megabytes of storage on a disk. When you rip a disk to your hard drive, do you end up with 650 megabytes of files? Not even close. I mean, an MP3 might be, say, 2 megabytes, right? So clearly there's a lot of compression going on. And that compression is taking place because the MP3 format realizes that there are some frequencies that you just don't hear so well. Even if you're as young as you are, it's true. So if you sort of ignore a little loss of fidelity in that frequency range, who's going to notice? Has anyone ever tried to make an MP3 from an MP3? A few people have. Well, if you do it, I think what you'll discover is that the most straightforward way that this happens is that a buddy rips an MP3 for you and then writes a CD, an audio CD from that MP3, and then you try ripping it again. What comes out sounds terrible. Just don't do this to your ears, it will hurt them. So the problem there is that in coming from the original CD to the first MP3, information was thrown out. And when MP3 encoding is applied on top of MP3 coding, what you hear sounds terrible. Frequently, you will find that people are sharing meme graphics on Facebook. Surely someone has seen something of this sort. Maybe it's a criticism of the president, for example. We see that stuff all the time. So, frequently, you will find that people have downloaded this image to their computer, saved it in another format, and reposted it. And what you end up with are what I call mosquitoes, these little trembly bits on the edges of text. So something like that is an example of lossy compression being applied on top of lossy compression. So ideally, in an ideal world, we should use lossy compression only once at that very last step. Okay, so the idea behind Jpegs lossy compression is that if you shift brightness, luminance information, our eyes tend to notice. So if we were to drop to 6 bits rather than 8 bits for intensity information, you would notice kind of a stair-steppiness about a grayscale shading. So that's not very good. We can't really shift luminance, but our eyes are simply not that good at detecting color. I mean, you would never think so in South Africa, right? But in fact, our ability to perceive color is pretty trashy. We've just got these 3 receptors, right? So, am I able to differentiate her sweater back there from, could I tell whether that is pink or salmon? I'm gonna go pink, but it could be salmon, and frankly, my eyes are not that hot. I might not notice, right? So we have the ability, if she were to leave the room, change sweatshirts, and come back, would I notice the color shift enough if it were a difference between pink and salmon? The answer is absolutely not. Absolutely not. So our eyes are not that great at chrominance, at what hue something is, or how saturated it is. Brightness differences, however, we're pretty good at. One of the reasons is we used to have to run from predators in the dark. So having luminance detection is really important to us. So every time we save an image to JPEG, we're going to reapproximate that image. So you start with a bitmap, you save the JPEG, well that looks pretty good. But then later on you decide you want to add a caption to the picture. You now reopen the JPEG, write some text on it and save it. Now when you look at it, it doesn't look that hot. It looked a whole lot better the first time. And the reason is each save is reapproximating the image. The image gets split to 8x8 blocks, and within those 8x8 blocks, the software uses this DCT compression to represent that block of 8x8 image. But what you get once you keep using DCT, the discrete cosine transform, is blocking, ringing, and mosquitoes. So blocking means that what was originally a nice smooth shading suddenly has stair steps in it. This one has like 4 levels of brightness where before it had 8 different levels of brightness. That's blocking. Ringing means that there are places where we have high contrast in our images, right at the edges of things, right? So we have, for example, the edge of her face in this image. So ringing is something where you get sort of a shadowing, where you don't have just one sharp edge at the edge of her face. Now you have 3 edges there, kind of a double chin, except having just a few pixels apart from each other. Mosquitoes are what happen when you, for example, save text, reopen it, add more text, and resave it. Now you have these little fuzzy bits showing up around these hard edges. So it's another way to talk about ringing, for example. So we use lossy formats all of the time. We use MP3s, we use JPEGs, we use QuickTime movies, we use even video for Windows AVI formats. All of these use lossy compression to a very, very high degree. All right. Now we're going to talk a little bit about complex data types. And the reason that we have to do this is that the bioinformatics uses these all of the time. XML is one of these formats that we use for lots and lots of data representations. They allow us to store very complex kinds of information in the context of a file. Sometimes you will see them with an XML extension, but not every file that uses XML is storing it in a file marked .xml. So I wanted to start with the example of HTML. HTML and XML are cousins, right? They're both examples of, let me see, is it the, I should know this off the top of my head. SGML is kind of the standard generalized markup language. From that we have HTML and XML as cousins. So many people have done some sort of editing on a web page, maybe by working in WordPress, for example, or of course creating your own Facebook page, whatever. Underlying all of this is some sort of HTML document that explains what text should be represented there. So here I've given you the complete text of an HTML web page. We start with a big tag that surrounds everything called HTML. HTML start and HTML end. When you have a slash in front of it, that means that's the end of this element. Inside that, we have a head element and we have a body element. See, the body element starts here and ends here. Within the header element, we have a title item. And this title says Dave's page. So when you render this HTML in a web browser, Dave's page is what appears in the title bar of your web browser. That's how that's set, by a title element inside the head of the HTML. Inside the body, I have some structure. I have a header 2. So I've decided that I want to have a big title at the top of my page. I could have gone with the head 1 element, but come on, we got to show some restraint here. So we used header 2. And I specified, Dave is awesome. That's my big text at the top of this page. Inside that, I have a paragraph element. Here's my paragraph right here. It starts at this, ends at this. Even if I split it across multiple lines, because it's all fitting inside one paragraph block, the browser knows this text should be kept together as a paragraph. This page is cool. Here I've got style tags. I'm using an italics item to say cool should be shown in italics. If I were really being a jerk, you know I would use the blink tag, right? We don't do that. That's bad practice. All right. And so this is the document. HTML works because the people who define what web pages should look like have set this definition for what tags are allowable. What if instead of writing, using the italics tag around cool, I had instead used my word spiffy, right? I've used my spiffy tag to mark cool. Does your web browser know how to render a spiffy tag? It does not, I think. I'm pretty sure that didn't make it into the W3C's canonization of what HTML should look like. So I am compelled to use only those tags that are defined by the W3C that are understood by your web browser. All right. Now at the right, I'm showing an example of MZML. MZML is an example of XML. So it's a subtype of XML. It's defined by a group called the Human Proteomics Organization Proteomic Standards Initiative, HUPO-PSI. And so here they have given information at the top of this file. This is just a very, very small subsection of a multi-megabyte file that is specifying on what kind of instrument were these data produced. So there's a referenceable param group list that's got 2 items within it. We see that the first referenceable param group is the set of common instrument parameters. Here they've got something called a controlled vocabulary that specifies exactly which terms can be used. So the mass spectrometer here is defined as a Q exactive. This is a kind of mass spectrometer. We see that there's also a space for storing the instrument's serial number. Q exactive series slot 542 is that. And so just this bit of text has a set of controlled terms that have been defined by the people who created this particular subtype of XML for storing bioinformatics available information. So if I have a whole pile of MZML files and I'm trying to figure out which of them were produced from a Q exactive, I could sniff out this controlled vocabulary term from the XML and grab out which files corresponded to that instrument type. This gives us the ability to store arbitrary types of information in XML. It also has the ability to store compressed information within the XML format. So you can do some pretty amazing data structuring with XML. Sometimes, however, it's very, very important to us that we're able to retrieve information very, very quickly from a very large set of data. And for that, we'll frequently make use of a relational database management system, RDMS, using a language called SQL. So RDMSs are commonplace. Most people, when they install Microsoft Office, install an RDMS along with it. Anyone ever use Microsoft Access? Okay. One person has used Microsoft Access. Well, Microsoft Access sits behind an awful lot of databases that are created in the wild. If you have installed Access on your computer, you have the ability to create them right there. So sometimes we will see that people are creating repositories for different tissue types that they have stored in their laboratories. And frequently you'll find that people do this in Excel, making big spreadsheets. And they have information problems with that they don't really think about. We'll talk about some of those in a minute. But if they had instead used Access, they would have the ability to do querying against these large logs of samples and be able to figure out which freezer they need to go look at to find a particular sample. There's a reason we have well-structured systems for this purpose. So IBM, back in 1970, I wasn't even born yet, E.F. Codd wrote this paper on scalable strategies that would allow us to retrieve information very, very quickly, called the Relational Database Management System. So all the way back in 1970, these things were given birth. IBM felt that a standard way to interact with RDMSs was valuable, so they created the SQL language that was later abbreviated. It stands for Structured Query Language, just so you know, in order to support the way we can manipulate these. So if you have a modern database, you can almost always talk to it using SQL. MS Access is an obvious example. There's one that frequently shows up under other names called SQLite. It's a very easy to use format for developers to integrate databases into their arbitrary software. MySQL is a very common one that we see over on the fine world of Linux, as is PostgreSQL. I think most people have heard of Oracle, very, very big company that's made a lot of money from marketing database systems. So one of the key things that you can do with this is to not deal with a single table, but to merge together information across many tables. So here we have a contact database for some company. We have a history over here that helps us to keep track of how we know people. We have different types that have been specified for this. Which staff member has made contact with this particular person, for example? Who is the contact that we're trying to store in the database? How can we communicate with them? What company does this other person represent? Company types are also spelled out here as well. So by having all of these different bits of information connected, we're able to ask complex queries. You might say, for example, I'm thinking of doing business with a poster printing company. Who do I know over there? So by querying out, by pulling out that particular poster printing company, we can get information about which contacts have been made at that company. Everyone sees that relationship forming between tables. Okay, so relational databases give us a lot of flexibility in modeling these complex structures. So we start with this idea of the index. The index is a really key concept within databases. So an index enables us to sort through a very large number of people at the same time and find only those that have a particular identification number. In the United States, we don't have a national identification number, for example. There's no citizen number or whatever. We frequently find that people use a fallback system, which is the social security number that we get assigned. So we're assigned a social security number that allows the government to track that we as employees have moved from company to company and we're contributing towards the retirement fund that we will be able to draw from late in life. So people end up using these social security numbers, not realizing that sometimes they're non-unique, that multiple living people may have the same social security number because the system was never made really bulletproof against errors like that. Okay, but if we can index people by some sort of identifying number, we can make it possible to grab out all information pertaining to them. So then we have this concept of an inner join. Combine the data across two different tables, aligning on a field held in common to both. So if you have one record for a particular identification number in one database, and that person has made multiple purchases with our company in another table, this inner join will create a new set where every order from this person is combined with the information about that person. Okay, so a query is this process where we interrogate the database to get information out. I didn't make this flicker white when I was tromping on the cables there, did I? Okay, so I'm going to stand over here. So when we do a query in SQL, it almost always starts with the command select. So select is a very widely used command within SQL. We're now going to create a new table that combines information across tables. So we're going to have the column ID and name and amount and date. So 4 columns drawn from customers. So customers is one of our input tables. we're going to build this information via an inner join that combines information from orders and customers, where in any case where the order's customer identification number equals the customer identification number. So in effect, this is pulling up an order history. We're going to pull up the list of all orders that were made by a particular person in our catalog. Great. I don't expect that you can speak SQL, but when you see the select statement, you should realize, I'm grabbing information from the database here. Okay, so normalization is a very, very important topic, and it's one of the, I think, the best reasons why using Excel for complex data storage is a really, really bad idea. So I'm going to try to walk you through this. I have a little demonstration table here. I have students in a school, oddly enough. So I have an identification number for each order, for each row here, 401 through 404. I have the name of a student, Adam, Alex, Stuart, and Adam. Well, this is obviously a sexist table, I'm sorry. We could just read this as Sarah, if you like. Sarah from Cape Town rather than Stuart from Cape Town. Is that okay? All right. We have a city in which that student resides, and we have a subject. Now, Adam is a little freak. Adam has decided that Adam wants to study not just biology, but also physics. What person likes both physics and biology, right? So, here's Adam. Now, I want you to remember that this Adam and this Adam are the same person. They have different index numbers here to reflect that different subjects have been listed as being of interest. Okay, so let's talk about the update anomaly. If we add a record, we have to re-enter existing data for consistency. Let us imagine that Sarah, rather than Stuart, we've replaced Stuart with Sarah. Sarah now adds a physics major on top of her maths major. So now we need another row in our table based on the way we do this. So 405, Sarah, Cape Town, physics. So to update the table with her new major, we needed to add information that was not changing from the existing record. After all, what would it mean if we had written 405, Sarah Stewart, Durbanville physics? Right? Now we've got this confusion that the table can't remember which city she's from because we've given two different locations for her. We don't know we don't have a way to establish that's right. So we have the potential to make a mistake every time we update our table with new information. We have an insertion anomaly. If I'm adding a new student here, let us say Jane. I add Jane here. I know that Jane lives in Belleville, so that's cool. Jane Belleville. But Jane hasn't picked out what subject she's going to study yet. So I have this problem of when I'm inserting a new record into this database, I don't have all of the information I need. I have an empty cell now, and I have to say, a lot of systems really freak out when you have empty cells. So designing the table this way is bad for the insertion reason as well. Likewise, deletion anomaly. When we remove a record, we lose more data than intended. What do we do if Alex decides he's no longer interested in maths? Now he's going to change his major, but he knows it's not going to be math, but he's not sure what it will be. Well, we have this problem that we can erase the entire record, but then we lose track of the fact that Alex is from Belleville. And so we have to have basically empty cells in our table in order to accommodate the student's change of information. So these are all reasons why doing a single table in Excel is a bad idea. And it also explains why some of the databases that we see in the wild are poorly structured. They're structured in ways that they leave us vulnerable to update anomalies, insertion anomalies, and deletion anomalies. This is a wildly complex topic, but if you understand these three anomalies, that I think you've got the first stage in understanding why databases are so necessary, why well-structured databases are so necessary. Okay, the time is 10:22. I have about another 7 or 8 slides. I can either power through or we can take a quick break here for students to stretch legs. What do you think? A little stretch? Okay, I'm going to ask you to hit the record button again, and we'll come back in just a minute. Okay, welcome back from break. I find that's very refreshing. Just to stop talking for a moment is a really good thing. So, I've drawn 3 diagrams back to back, and they've been included in your slides. They are fairly complex, but I thought it would be useful to try to illustrate the diversity of topics that fall under bioinformatics. What I'm showing is a huge number of different algorithms that have been created in the space of bioinformatics that cover all kinds of different goals. So we're going to start with a few on a few of the very active bioinformatics research areas within sequencers and microarrays. From there, we'll move on to talk a little bit about some of the methods that have been used in proteomic and metabolomic assessment, and then we'll talk about biological pathways and networks. So I mostly want you to understand how broad the set of goals are that we contemplate when we think bioinformatics. So I would note that sequencing is not sequencing. You know, the sequencers that we used back when I was in grad school were largely limited to 96 different templates that we were trying to measure at the same time, that we're trying to sequence at the same time. And today, even a very poor old school next-gen sequencer will sequence millions of different sequence templates at the same time. So all of these technologies are continually in renovation. A lot of people think of microarrays as very old school and not very up to date, but in fact the amount of information you can get from them is still pretty dramatic. So I've tried to knit together where microarrays touch sequencers. So I've listed topics of genomics and transcriptomics, but I note that there are arrows that lead between these two because sometimes the experiments that we carry out via RNA-seq, for example, in transcriptomics, can be repurposed to look for genetic variants. So just because one has done an experiment of one type doesn't mean that it can't be analyzed in another. So I've really separated these into a problem of novel genome annotation. What happens when you sequence the honey badger, for example? You know, you want to make sense of all of this genomic sequence that you've gotten back. The second trace is really more around the concept of resequencing, that we have already sequenced what the human genome looks like. We know very well where the genes are, but now we're sequencing a bunch of new people in order to understand population genetic variants in an attempt to understand which genetic variants have which relationship with phenotype, for example. So on our first track, dealing with a novel genome, I've written as the output a functional annotation, learning which bits of genetic code do what. At the right, I've sort of got the concomitant output, the genome-wide association study, for finding which bits of genetic variation are mappable to particular phenotypes or give us a boundary to that. Now over at the right, under transcriptomics, kind of the obvious output from a transcriptomic study is a set of genes that are known to vary in response to some phenotype. Not to vary in their sequence, but to vary in their expression level. Basically, how much do we hear this gene when we look at the transcripts that are produced from it? Bioinformatics is all over this. So things like base calling are quite essential when you're dealing with any type of sequencer. Being able to determine what letter appears at what position in a genetic read is one of the most fundamental things that we must do anytime we use a sequencer, whether it's a Sanger, old school gel or capillary electrophoresis sequencer, or one of the much newer next-gen sequencing class instruments. Base calling is essential for all of those. Being able to do high-speed mapping means that we need to be able to figure out where in the human genome this particular read that we have produced for this person belongs. When we have thousands and thousands, or actually millions of different reads that we need to map against a very complex genome, high-efficiency algorithms are certainly necessary. So one called the Burrows-Wheeler alignment, the BWA alignment, has been a very popular one for this purpose. When we do assembly of large genomes, when we have a bunch of reads and we don't know how they all fit together, finding automated ways to deal with whole genome shotgun data to assemble contigs or whole chromosomes even from these reads is an essential computational task. And this is, I think, one of the really huge advances that have taken place in the course of my career for being able to do large-scale de novo assemblies. How do we find genes, right? If you've just got megabase after megabase of DNA, how do you figure out where the bits that actually correspond to protein coding genes are? It's a very hard task. So we have developed systems like hidden Markov models that are very good at spotting things that look like genes. And we have people working here at UCT actually who are very valuable, who have made very valuable contributions in the space of recognizing that we think this is a gene, so what function does it have? How can we align this, this protein sequence to all kinds of different signatures that are associated with biological function that have been observed in other sequences? The whole interpro system, for example. So I think Nikki Mulder will probably visit with you at some point. She's a real expert in that space. So being able to do base calling and read mapping is great. Next we need to have statistical and systems that are appropriately weighted for recognizing that this letter in this read is something we can trust as a genetic variant that this person features. When you're dealing with data from both chromosomes of that person, can we spot that a person is heterogeneous for the alleles that they have at this particular gene? So being able to do variant detection very well is a critical capability. Being able to deal with admixture analysis is quite important anytime we do population studies here. South Africa has some of the most interesting population stratification in the world that can make it really challenging to figure out which genetic variants are associated with phenotypes. There's a big study that came out of my university, Stellenbosch University, that was looking at the population genetics of the Cape Colored population, trying to understand what adjustments must be made to know that this genetic variant that we see is not just because of co-segregating alleles, but is rather truly associated with the biology of this phenomenon. It's really quite challenging to do this. In the United States, a lot of the genetic medicine approaches that we would like to use to support the African American population, we actually can't because the alleles that we see in the African American population are different, so we don't have as much information about which alleles are significant in disease. And for each individual, their genealogy is going to be this unique combination of African and European alleles. So unless you untangle that structure first, your ability to determine the meaning of genetic variants that you see is much limited. So South Africa runs into quite the same situations. So yes, this admixture analysis is a critical part of this that has to come into play anytime we deal with genome-wide association studies. Base calling and read mapping are still essential in the world of RNA-seq, although we use slightly different methods in the context of RNA mappings to genome than we do in exome sequencing. But being able to recognize novel boundaries between exons that we haven't previously seen being spliced together in messenger RNA matters, certainly. There's all kinds of normalization and quality control assessment that we need to do in the context of microarrays. These are concepts like biclustering, recognizing genes that are up and down in the same people, in the same patterns, and recognizing people that have gene expression profiles that are similar to each other is a very valuable bit of information. These last three I've shown in green, not that we could tell without a blue lamp in the projector. And these are some of the most statistically important problems that we encounter in differential gene expression, being able to recognize genes that are that we can determine robustly are truly differential between different cohorts. So these are just some of the ways, just within the sequencer and microarray world. Things get pretty diverse once we start thinking about the proteomic and metabolomic world. So with proteomics, we have kind of a split world in that we have people who think about protein identifications, protein quantitation, and post-translational modifications, who are mostly dependent on tandem mass spectrometry as their data source. But the other half of proteomics is this fine world of structural biology. Understanding the structure of something is really critical. The way that we think about it is that sequence should give us almost all of the information we need to understand why something will adopt the structure that it does. If we know what structure something has, we can probably associate a function with it. So sequence to structure to function is one of these rigorous things that we always try to work our way through. So people who are trying to measure structure are probably using X-ray crystallography or NMR, or are using electron microscopy to try to analyze these structures, each of those at sort of a decreasing level of detail. So if you are dealing with tandem mass spectrometry, being able to identify peptides and proteins, being able to control false discovery rates, being parsimonious about which proteins we invoke, being able to deal with post-translational modifications and recognize where they fall within a particular protein, these are kind of the canonical things that we see within the proteomics realm, and something that Dr. Blackburn is quite good at. And from there, we have kind of the same problems. Again, these are shown in green, not that we could tell, to say which proteins differentiate these cohorts. Before we were asking, how does gene expression differ between two cohorts? Which genes are changing? Here we are looking at which proteins are changing, and very much the same statistical problems arise from that. Now, I mentioned the structural people are trying to directly measure protein structures here, so being able to solve the data that come back from their synchrotron or whatever becomes necessary, and that can give you a measured protein structure. Certainly, there's a very active world, though, that's working on being able to predict a protein structure just starting from its sequence. That's a really, really hard problem to get into, and although I draw it as just one box, I want you to keep in mind that that's got a whole lot of different flavors to it, from threading to just Monte Carlo systems for projecting structures and scoring them. Once you have a protein structure, you might want to know if we modify this letter of sequence from a valine to a leucine, does that in fact impact its function? Does it change its affinity? So for that, you might do mutation impact prediction. You might work on docking ligands to say which members of this combinatorial chemical library can be docked to this protein structure, to its active site. So that might give us the cue to particular drug leads. Or you might be trying to figure out which proteins interact with each other. So docking them to each other might help you to recognize, these have a really good binding interface, that their energy is really great for creating this interface. So protein-protein docking is another big interface. Getting the protein structure does not mean you're done. It means you're starting with a whole new round of informatic approaches. The metabolomics space has in large part trailed behind what's happened in proteomics and transcriptomics and genomics. In effect, you can think of them as a historical progression. Our ability to deal with genomic sequence sort of came about and was mostly in place in the 1990s. Our ability to deal with gene expression took a huge step forward with microarrays, and then again with RNA-seq. Proteomics has been sort of playing tail end Charlie on those. So of the three sequence-based disciplines, the climate for proteomics is kind of the least developed of those three. But metabolomics is still kind of in the Wild West zone, frankly. We have pretty good ability now to deal with quantifying particular metabolites. With that, we're frequently talking about something called a chromatogram. This is kind of a measure of intensity as a function of time. So if you have a particular time that your separation knocks off a particular metabolite or a particular peptide from the column, you're going to get a pattern of intensity rising and then falling off as this peptide or metabolite arrives at your detector. So these chromatograms frequently have variations in their times of arrival, so we need to be able to align for those effects. We need to be able to do peak integration to turn that chromatogram into a quantity that we can use to associate with it. That might in turn go to this differential pathway that we talked about, being able to recognize which metabolites now are differentiating to cohorts. Or it might flow into something called flux analysis, where we're trying to represent a biochemical pathway as a series of equations and ask how can we optimize the output of this pathway down this channel, not that channel, by altering its inputs. So flux analysis is really where you gotta go if you're gonna be one of these really deep math people. Systems of equations to model biology. It's a wild place to be. Being able to identify metabolites has been somewhat more limited. The major approach that we see there is something called spectral library matching. So I'm obviously not going into a huge amount of depth on any of this, but when you think bioinformatics, I want you to think that it's the essential handmaiden to all systems biology. Whatever pathway you're taking through systems biology, you're going to touch bioinformatics. And so understanding what is available to you across that space is valuable. All right, finally we get to biological pathways and networks. We have a very wide variety of ways that we discern which genes or gene products, or metabolites for that matter, are most related to others. Just for example, under microarray correlations, one of the things that we ask is when we've run microarrays of many different tissues under many different stimulations, we get some ability to parse which genes go up and down in connection with others. So this leads to sort of a guilt by association, right? If you hang around with somebody who steals chalkboard erasers, and if you're always hanging around with this person and a chalkboard eraser goes missing, somebody might be asking you where the chalkboard erasers are going, right? So in the same way, we ask which genes are likely to be associated with these other genes, gesundheit, just because they go up and down together. So this correlation leads us to believe that there's a relationship among those genes. We have lots of different methods that we've used for this. Each of them have their pluses and their minuses. Some of them are quite prone to false positives, for example. So we try to distill this information into a set of relationships among genes, gene products, or metabolites. From this, we build networks and pathways. Now, I've sort of specified gene ontology as something different than a pathway setup. That's not really fair, but ontologies combine information about shared relationships among genes with a controlled vocabulary. I mentioned that term earlier. This is a case where we have a set of defined meanings that we are allowed in this universe to use. So combining a controlled vocabulary with relationships allows us to build an ontology. And Go, gene ontology, is a hugely popular one in this space. So it kind of gets its own mention for that reason as well. These systems give us the ability to recognize genes that are more closely related than we would have anticipated. It allows us to walk from one part of a network to another to assess the relationship between things, to visualize, certainly. The fellow that I went to grad school with at the University of Washington arrived with a master's in mechanical engineering that he'd gotten from MIT. And I was always worried that I wasn't good enough because he was so sharp. Well, after he left grad school, he went to the University of California, San Diego, and created the lab that created side escape. So, my inferiority complex is just going to have to continue, I'm afraid. He really is that good. Trey Eidecker is a very smart dude. So side escape visualization gives us the ability to project molecular data in the context of molecular networks. The ability to do dimensionality reduction is a hugely important task in informatics. It gives us the ability to reduce our world from thousands and thousands of genes that we might be thinking to more tightly knit sets that relate to each other in terms of biology. This is really important. Whenever we use systems biology, sequencing, microarrays, mass specs, it doesn't matter much. We get back long lists of things. And being able to understand long lists is not really particularly good in our heads. Now, I'm just going to share a dirty secret with you. A lot of your PIs, if we give them a list of 20 genes that we say we have found to be differential in a particular circumstance, if we give them a random set of genes and tell them that we got them from a carefully designed experiment, what is their interpretation of this gene list? Almost in every case, they will come back to you with a biologically plausible sounding explanation of how these genes relate to each other. We have to protect PIs from doing that. And so one of the ways that we do that is through dimensionality reduction, using statistics to tell us which pathways are significantly represented, or using non-parametric systems like the gene set enrichment analysis for telling us which pathways or modules are significantly overrepresented. Biologists like to form stories from lists of genes. Someday, you might be a professor yourself. And if you are, you need to protect yourself against this. Be aware that your mind will tell a story when given a bunch of disparate items. Imagine if I walk up to a gifted storyteller and say, I have a feather, an egg, and I have a gold brick. What do these have to do with each other? That storyteller will probably tell us something like the story of the golden goose. But a PI is very much in the same position, and PIs have to protect themselves from being given disparate information and stitching a story together that has nothing to do with the biological reality behind it. Okay, so we're going to take a very quick blast then through some of the file formats that we see in the world of high throughput sequencing. If you haven't noticed already, I have a bit of a phobia of the term next-gen sequencing. Next-gen sequencing was out of date at the time it was coined, and a decade later it hasn't gotten any better. So I tend to think of this as high throughput sequencing. But there are other suggestions. We might call this, for example, massively parallel sequencing, because that's really the principle that seems to conjoin all of these new methods that we have for sequencing. They are powerful because they sequence millions and millions of templates at the same time. That's a huge difference. So an electropherogram is similar to a chromatogram that I talked about just a moment ago, a pattern of intensity in response to time. But here, we're looking base by base across a sequencing read. This is one reaction from the sequencer, and we have 4 different dyes, which would be visible if we had all of our colors here today. But we've got black, red, blue, and green. And we've got these different traces. So reading a sequence, trying to infer what the sequence should be for this read, we would say, well, that's a green, that's a blue, that's a black, that's a black, that's a black, that's a green. I think that's a blue. I really can't tell without the blue turned on. But we have these signals then that we're trying to interpret as letters of nucleotide sequence. And that's building from an electropherogram. These days, most modern sequencers don't report chromatograms. And the reason is simple. It's way too much information to store. If we store all this information, our sequencing files are huge. So instead, we report information from sequencers in this FASTQ format. It's just a big text file, essentially. So you'll have one line of text that has the read, the base calls for every letter across this read. And you'll also have another vector of characters that represent quality assessments. So the ASCII code of these characters tells us what the Phred score is for each of those letters. So there's already quite a lot of processing just to get to the FASTQ file. This interpretation of the electropherogram has already taken place, and an assessment of the per base quality has already been made before you get the first bit of output from the sequencer. It's worth remembering, a lot of processing going on there. Being able to report alignments is a hugely important task for us. If we've done an RNA-seq experiment, we need to know which transcripts have, which exon-exon boundaries have been observed. Do we have the ability to quantify how much this isoform rather than that isoform has been output? So what we see frequently are the SAM and BAM files. So sequence alignment maps store the report that says we had this. You can see a lot of different roles for SAM files. You might, for example, have a novel genome where you've built contigs from these reads to represent the parts where reads can be found overlapping each other. So you might have a list of contigs that have come out of a de novo assembly, or you might have a mapping where you have a known reference that you've mapped reads onto. So SAM and BAM play this very essential role. SAM and BAM represent a lot of heavy processing on the back end of those FASTQ files that we talked about. The FASTQ is the output from the sequencer. SAM and BAM is our alignment built from it. And then frequently you'll want to visualize particular data within a genome. Maybe you want to show how a set of reads mapped to a particular transcript. So for something like that, using a browser extensible data file, a very compact file, may help you to store all of this information for visualization in an external framework. There are lots of visualization systems out there, like the genome browser from UCSC, for example. Okay. So we also do a fair amount of highly distilled information. One of these examples comes from this world of genetic variation, trying to look across a population and say what genotypes do we see. So being able to use, one of the inputs from this is typically the general feature format, which gives us the ability to store our annotation in something more intelligent than just a FASTA file. Right? You can download a FASTA file of all of the transcripts for the human genome. That's fine, but that doesn't give you information like which exons at what genome coordinates are getting pasted together to create this transcript. For something like that, you would want the general feature format, the GFF file. This has gone through several revisions over time, of course. Or it may be that the only thing you actually care about are specific variants that we're seeing across many genomes. So for something like that, you might have a very compact output called a VCF, or a VCF. So this is just calling out basically an individual's genotype. Of course, some of that information must be retained, must be withheld to protect the privacy of people who take part in biomedical research. So we have to always be thinking about people's privacies there too. These URLs that I've given at the bottom certainly can give you a lot more information about what these formats are used for. So that's a lot of information, I appreciate. I've been rattling on for very near 2 hours, so I'm going to call it to a quit right here. But in closing, systems biology employs bioinformatics for initial pre-processing, follow-on summarization, established knowledge representation, and visualization. If you are doing anything in systems biology or in chemical biology, you are going to be using bioinformatics, of course. And so hopefully this has given you a little bit of a running start for recognizing some of the things you're going to encounter in that search. Bioinformatics draws upon diverse computer science fundamentals to accomplish its aims. I've really just scratched the surface. You can't do much more than that in 2 hours. But I hope that when you encounter some of the different representations along the way, you'll recognize, oh, we talked about that. Biologists must understand some computer science. Computer scientists need to learn some biology. The second part may not be that intuitive to you, but in fact, a lot of the most interesting problems that have challenged computer science in recent years have come from molecular biology. Computer scientists frequently come to us asking about interesting ways that they might apply their algorithms because we have the cool data. You know, we're a long way from running through fields with butterfly nets, right? The type of data we can harvest from an individual sample today, even if you've got just a single cell, is pretty remarkable. So I'm glad you're joining this field, and I look forward to what you guys are all going to accomplish. Thank you.